Okay. Testing. Alright. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh dan selamat pagi. We have 13 people here, but in the paddock there's only 11. I would like to encourage you to join the link for paddock as attached in the chat or in your WhatsApp or in your Telegram group, because all the notes that I've given and I will be using throughout today's class will be automatically sent to you by paddock when I click on the prompt. So tak adalah isu nanti ada orang tak dapat notes ke, nak minta notes kemudian ke, sebab automatically akan dapat. During our class, for those of you who are familiar with the way I conduct my class, I will be using this app in order to elicit responses from you, to get your responses, because some explanations atau the way I deliver my content is also depends on how much you know about certain topics. So I will adjust the way I explain, or how much should I explain, based on how I gauge your knowledge based on your responses. So please join the paddock app. Kalau ada masalah kalau ada masalah internet connection, sampai you have to choose either Google Meet atau paddock, then I understand if you don't join the paddock. Okay, but if you can, please join the app, because it will send the notes that we'll be using today automatically after class. Okay, that's number one. Number two, we will be discussing about SBP paper 2. When I take a look at the content of the paper, and the time that we have, I think the only part that we can discuss is part A, bahagian A, all of the 5 questions. Itu pun saya select, dan ada yang saya ubah suai dia jadi soalan soalan multiple choice question, so that we can save time. So let's see, hopefully we can finish all 5 questions today lah. Alright, mari kita start. Thank you so much for all of your beautiful drawings. This is the way I gauge whether you are having a good internet connection or not. Sebab boleh lukis ni, okay lah, maknanya and then I want you to familiarize with the app. Number one, even though this is a drawing slide, you can also use text. You can also use text for the next parts of the paddock app. Nanti kita ada banyak kena tulis jawapan, kena lukis. Jadi it will be good if you can also try the text tool at the bottom of the screen dalam paddock. Okay, let's start. Okay, I give you 1 more minute to finish your drawings, finish your masterpiece, and we start the class on 10:05. Okay, it's already 10:05. Mari kita mula. So today we'll be discussing about SBP paper 2. SBP paper 2 ni is one of the questions that is provided in your module, yang kita bagi hard copy soalan-soalan tu ada dalam tu. So kalau ada yang terlepas ke atau ada gambar yang tak clear ke, you can refer to your hard copies. So a quick analysis. A quick analysis on section A, we can see that it started off with movement of substances. Selalunya the first question dalam bahagian A ni ialah chapter 2 iaitu cell structure and organisation, tapi dia start dengan movement of substances and then proceed with cell division and plant reproduction. Memang both of these go hand in hand. Kalau cell division mitosis selalunya dia associate dengan growth. Kalau cell division meiosis selalunya dia associate dengan formation of seeds ke, formation of ovule ke, formation of formation of pollen ke dan sebagainya lah. And then the next part is chemical composition, dan chemical composition of the cell ni memang part of it ialah describing factors affecting enzyme lah. And then it ended with coordination and response, specifically human nervous system and inheritance and variation. Inheritance variation ni dah jadi a staple in section A question 5 sebab soalan dia memang soalan dikira sebagai aras tinggi lah. So ini analisis untuk section B, which we will not cover today because of lack of time. So we will focus on section A only. So this is the first question. I've already given to you what is P, what is Q, and the name of the process, and then can you help me to explain how the processes occurs? Can you help me to explain how the processes occurs? Use text, boleh guna text. So when we explain how the processes occur, we started off with nyatakan dulu what is the difference of concentration atau what is the difference of partial pressure. Kita sebut partial pressure sebab kita cakap pasal gas. Tapi if you use concentration pun okay. Okay. This is an application of simple diffusion. Macam mana kita nak cerita simple diffusion? Start with siapa yang concentration atau partial pressure dia lagi tinggi atau lagi rendah, and then followed by oxygen atau carbon dioxide. Oxygen will diffuse from where to where? Kenapa kita tak cerita carbon dioxide? Sebab rajah tu dia tak label carbon dioxide. So kita tak payah cerita lah. Kita cerita pasal oksigen saja. Okay, you try. Mesti cuba at least, mesti jot down, cuba, so that you are forcing yourself to produce ideas, what you have in your mind. Kita keluarkan, dan bila kita tahu, oh rupanya yang kita tahu ni tertib dia betul, ataupun yang kita tahu ni tak cukup, jadi kita akan lengkapkan dekat situ. Alright, so these are some of the answer. Okay, so some of you started with naming the process, which is good. Tapi untuk soalan ni, sebab soalan 1A1 dia dah bagi tahu nama proses. So if you mention one more time the name of the process dekat soalan sekarang ni, tak dapat markah lah. Sebab soalan sebelumnya tu dah bagi tahu, dah minta berapa. Okay, so here are a few examples of answers. Okay, so boleh start keluarkan pen merah cikgu-cikgu dan boleh tanda atau highlight part mana yang memberikan markah. Okay, so number 1, kita bagi tahu dulu the difference in concentration or partial pressure. Number 1, partial pressure of oxygen in alveolus is higher compared to blood capillary. Kalau nak guna concentration pun boleh. The partial pressure of the concentration of oxygen in the alveolus is higher than the concentration of oxygen in the red blood cell pun boleh, tak ada masalah. Okay, that should be your P1. Tanda dekat situ P1. And then let's take a look. Okay, the next one is partial, sorry. So oxygen will diffuse from alveolus into blood. Okay, so oxygen will diffuse from alveolus into blood. Okay, bila kita nak cerita pasal simple diffusion, kita just cerita nombor satu, perbezaan kepekatan, difference in concentration. Siapa lagi tinggi, dekat mana lagi tinggi, dekat mana lagi rendah. And then kita cakap molekul yang terlibat iaitu oksigen akan diffuse dari mana ke mana. Kalau kamu sebut move, tak dapat markah. Sebab diffuse is the verb that we will be using specifically for this type of process. Okay, thank you for your responses. Okay, boleh dapat markah 2 kat situ. Okay, now explain the characteristic of P which is related to polarity. Okay, so P is phospholipid dan saya bagi gambar phospholipid kat situ. Markah dia 2, senang saja. Apa 2 karakteristik dalam phospholipid, pada phospholipid. Okay, you can start to answer. Okay, so memberikan nama kepada structure tu pun tak ada masalah, boleh. But because the previous question already ask you to label P tu apa, Q tu apa, tak dapat markah untuk nama lah kat situ ya. Tapi tak apa. Okay, so P sifat dia, karakteristik dia ada 2, iaitu dia ada hydrophilic head atau polar head. Polar head pun kita terima ya. Hydrophilic head or polar head, and hydrophobic tail atau non-polar tail. Okay. Hydrophilic, hydro means water. Philic means suka, I like. File kan, file. Macam thermophilia. Thermo means heat, philia means suka. So hydro means water, philic means suka. Phobic comes from phobia. So hydrophobia, takut air, tak nak kena air. Alright. Dapat 2 marks kat situ, senang saja. This is a very easy question. Okay, mari kita tengok soalan berikutnya pula. Soalan berikutnya sebenarnya dia minta kita draw the condition of one of his red blood cell. Situasi dia sekarang ialah bila kita mengalami diarrhea, kita cirit berit. Bila kita cirit berit, kita akan banyak keluarkan air daripada badan. Kan sebab bila kita cirit berit tu kan najis kita cair kan. So this is because makanan yang tak dicernakan tu lalu dalam usus besar dan lalu dalam kita punya kolon dengan cepat, atau kolon kita dalam keadaan sedang membengkak ke, ada infection ke. Jadi kita punya kolon atau usus besar our large intestine cannot absorb water from tak payah lukis ya sebab dah lukis dah. Explain how this process occurs. Lukis dah ada dah. Saya dah bagi gambar kan. Okay. Nak soalan lukis-lukis je. Sagi kita ada jawapan lukis-lukis. Okay, saya ulang sekali lagi. Bila kita mengalami diarrhea, kita akan mengeluarkan excess water daripada badan sebab najis kita cair kan. This is because our large intestine cannot absorb water because it is infected ke atau ada inflammation ke. So a lot of water cannot be reabsorbed from our food substances, reabsorbed back into our bloodstream. So this is why this causes the lack atau decreasing volume atau decreasing water content dalam plasma darah. Dalam larutan darah tu kurang air sebab tadi tak ada air reabsorb atau kurang air reabsorb daripada digested food and digested food dalam large intestine masuk ke dalam darah. Okay. So situasi dia ialah sama macam kita nak cerita diffusion of gases tadi. Sebab markah dia markah dia 3 saja, kita kena fokus kepada proses osmosis. Soalan tadi tanya simple diffusion. Soalan ni dia tanya pasal osmosis. So number one, bila kita cerita pasal osmosis, kita cerita siapa yang hypotonik atau siapa yang hypertonik. The words. Siapa yang hypotonik atau siapa yang hypertonik. If you are talking about red blood cell, so adakah red blood cell tu hypotonik ke, hypertonik ke, isotonik? Compared to the solution outside. Solution outside red blood cell tu ialah blood plasma lah. Macam tu ya. Okay. Okay. So surrounding solution atau yang lebih tepat lagi plasma darah lah. Red blood cell is hypotonic. Good. Okay. Okay, hypotonic compared to blood plasma. Itu baru point 1. Okay, point 1. Point 2. Jadi, kita cakap pula pasal diffusion of water molecules. So, anak-anak bila point yang saya sebut ni ada dalam kamu punya jawapan, kamu highlight dan tanda ya. So, explain how this condition happens. Dalam gambar tu, diagram tu kita lukis red blood cell sudah cremated. So, when we talk about bila istilah dia cremated atau description dia crenation, kita tahu ini adalah proses osmosis. So, we are describing osmosis dalam soalan ni. When we describing osmosis, the first point is to talk about which one is hypotonic, which one is hypertonic. So kalau kita cakap dalam konteks red blood cell is hypotonic towards blood plasma. Okay, that is point 1. Point 2, water molecules diffuse from where to where? Water molecules diffuse from red blood cell to blood plasma. Okay, ada yang sebut water from red blood cell will diffuse out to the blood plasma. Okay, via osmosis. Good. Point number 3, red blood cell become cremated. Atau red blood cell will shrink. Red blood cell shrink. Okay, 3 markah. Siapa yang hypotonik, water molecules diffuse to where, and then the condition of the cell. Kita tadi lukis saja keadaan sel, tapi kita tak bagi nama kepada condition tersebut. So boleh dapat markah. Alright. Boleh tanda right ya. Tanda right pada diri sendiri. Boleh tanda right pada diri sendiri, jangan lupa. Boleh tanda right. Nanti bila kamu semak kamu punya jawapan, macam Aimi ni dia highlight kat mana yang dapat point kan. So nanti bila kamu semak balik jawapan kamu, kamu tahu. Kamu semak balik nota ni, kamu tahu part mana yang memberikan markah. Alright. Let's go to the next one. Next one. Okay, untuk yang ni saya ubah suai soalan ni kepada soalan pilihan. Okay. So soalan ni sepatutnya kita state the characteristics of meristem cell. Kita belajar, kita dah kenal itu adalah meristem cell sebab itu adalah yang dilabel dekat situ ialah kawasan apical meristem where we can find meristem tissue. So meristem tissue kalau dia tanya type of cell dibina oleh meristem cell lah. Kenapa? Sebab kita dah belajar the same type of cell will combine with each other to form tissue. So kalau kita tahu kat situ meristem tissue, maknanya kita tahulah dibina daripada meristem cell. Okay, so you have 4 buttons there. Choose which statement, ada satu, dua, tiga, ada 8 statement kat situ, hanya 4 saja yang betul untuk meristem. Hanya 4 saja yang betul untuk meristem. Cuba pilih. Most of the characteristics can be observed from the diagram. Can be observed at the diagram. Okay, characteristic 1. Okay, characteristic 1 ialah small vacuole. Siapa yang pilih big vacuole tu salah. Kena tolak kamu punya ikon, pergi dekat small vacuole. Cuba tengok dekat rajah tu betul-betul. Nampak ke vacuole kat situ? Tak ada kan? Okay, siapa yang letak dekat big vacuole tu, tolong alih ya, tolong alih. Syafika, siapa ni? Mikael kot ni. Okay, Khalil. Okay, thank you. Thank you for your prompt action. Okay, number 2 ialah nukleus dia besar. Big nucleus. Big nucleus because it is constantly atau repeatedly conduct mitosis. Okay. Number 2. Okay, big nucleus. Ada seorang yang masih dekat small nucleus tu, tolong alih. Okay. Okay, next ialah dense cytoplasm. Dense cytoplasm. Because the nucleus is so big, it pushes all the other organelles ke tepi. So kita nampak cytoplasm tu dense lah. Semua organel terpaksa berkongsi tempat. Okay, and then thin cell wall. Cell wall dia thin saja. Nipis saja cell wall dia. Okay, kalau kita tengok pun dalam gambar rajah tu, in the diagram, segaris satu line saja dia punya cell wall tu kan. Dia tak nampak macam xylem tebal ada enapan lignin. So ada saya dekat saya punya dashboard ni, saya nampak ada lagi 4 student yang masih lagi dekat thick cell wall, alihkan jadi thin cell wall, tolak ke kanan. Thick cell wall tu salah Alright, you got it. Now, thank you so much. So these are the characteristic of meristem cell. Tapi dalam soalan dia minta 2 saja. So mana-mana 2 daripada 4 yang kamu pilih ni. Alright. So, kita tengok pula soalan yang seterusnya. Soalan explain balik. Okay. So, explain the chromosomal behavior at stage S. Okay. Explain the chromosomal behavior at stage S. Mulakan dengan apa nama stage tu? Apa nama stage tu? And then, what happens during that stage? Centromere buat apa? What happens during that stage? Centromere buat apa? Specifically, centromere buat apa? And then, what is the type of chromosome involved? Masa tu kromosom pada, nama kromosom pada fasa tu apa? Sister chromatid ke? Chromatid ke? Bivalent ke? Okay. So number 1, name of stage. Number 2, what happens? Apa komponen dalam tu sedang buat? Nombor 3, the name of the type of chromosome involved tu buat apa? Okay, kena ada nama. Masa tu ialah sister chromatid ke, chromatid ke, bivalent ke. Masa tu istilah tu kita nak tengok ya. Okay. Okay. Angka dia jawapan ni soalan ni ada 2 markah, mungkin ayat dia sedikit panjang. Jadi guna teks guna teks. Okey. So tanda jawapan kamu, tanda jawapan kamu number 1, nama stage tu ialah anaphase. So gariskan jawapan kamu atau tandakan, gunakan pen merah ya cikgu-cikgu, tandakan. Okey, nama dia ialah Nafis. Okey, nombor 2, poin yang kedua. Kalau tak ada poin ni pun tak apa sebab dia ada 3 poin, mana-mana 2 daripada 3. Point number 2 is centromere divided into 2. If you don't have it, it's okay. There is another point. Okay. Point number 2 is centromere divided into 2. Okay. Number 3, sister chromatid separate. Istilah sister chromatic tu kena betul. Kalau kamu sebut kromosom separate tak dapat markah Sister chromatid separate and move towards opposite pole. Okay. Sister chromatid separate and move towards opposite poles. Okey. Berikan tanda right pada jawapan-jawapan kamu ya, sebab nanti bila kamu semak, bila kamu dapat nota ni, kamu tahu and you can be proud of yourself. Yes, I got this correct during class. I can remember this part during class. I am okay for this particular subtopic. Okay, it's a good reflection for you so that you know which subtopic that you are actually good at, which subtopic that you have to work on, work more atau learn more. Okay. Ada seorang ni dah tulis S is Anaphase. Dia tulis tangan. Bagus. You got 1 mark kat situ. Bagus. Okay. Let's take a look at the next question. Let's take a look at the next question. Okay. These statements, okay, among these statements, which ones are true for process S? So there are 8 statements, only 5 are true. So which one? Okay. Which one ya? Ada 5 saja statement yang betul. Lagi 3 tu tak betul. So hanya bawa ikan kamu berenang ke statement yang betul saja. Renangkan ikan kamu tu, bawa dia pergi berenang. Okay, bawa pergi berenang dan pergi ke mana. Ada 3 statement yang salah. Yang hot lain tu 5 tu betul. Hot mana se. Okay, good. Okay, for those of you who just join in, please make sure, kalau kamu tak nak respon pun tak apa, I understand because some of us have device limitation. Please make sure you join the PEDAC app because right after this class, at the end of this class, the app will automatically send the notes for you. For each question, I've provided additional diagram, additional points to help you to answer and to help you to revise. So it will be good if you join the PEDAC app. Kalau jadi leka yang tengok saja jawapan pun tak apa, saya faham sebab because some of us have limitation in internet connection or devices kan. Okay. So let's take a look. Let's take a look at your answers. Okay, statement yang salah. Saya habaq my statement yang salah lah. Ada 3 saja yang salah. Okay, the first one that is not true is for producing seeds. Seeds of course lah salah sebab for producing seeds, it is the job of a flower structure. Tapi gambar ni tunjuk apical meristem. So it is not the statement for producing seeds tu salah. Okay, number 2, produce cell with haploid number of chromosome. Itu salah because haploid number of chromosome ni fungsi untuk proses meiosis, whereas we can see that S is a part of mitosis. So bila saya sebut it is a part of mitosis, sepatutnya statement yang terakhir yang confirm not true tu kita tak pilih, iaitu genetic variation. Salah genetic variation tu. Itu kerja meiosis. Itu tugas meiosis. Okay. Bukan mitosis ya. Sebab kawasan apical meristem ni dia mainly nak buat mitosis. Okay. Oh, so ada two people here, kejap ya. Two people here still letak parking dekat Fahana ni kot. Ada masalah connection kot. Oh, Fahana dah. Okay. Muhammad Ali, Muhammad Ali. Kejap, Muhammad Ali pula. Ada satu ikan kat sini ya. Ikan ni masih berenang dekat genetic variation. Tolong tarik ikan tu pergi tempat lain. Alright, thank you so much for all of your responses. We are halfway through. Let's take a 2-minute stretch break. Buat regangan untuk jari-jari kamu tu. Yelah kan, tadi dah lukis-lukis dan sebagainya. Tengok kat luar, tengok dekat tingkap kat luar tu. Kalau ada pokok, ada rumput. Tengok rumput. Green is good for you. Okay. Oh, dia pergi restart pula 2 minutes tu, tak apa. If you have any questions, you can ask. Dia tak ni pun ya. Okay. 10.33 kita start ya. Rupanya timer ni dia ada Google Slide ni dia limitkan hanya boleh play this particular GIF for 10 seconds. Alright, it's okay. 1 more minute, 10.33 kita start ya. Alright. Do you have any questions anak-anak? We have 47 people here. Do you have any questions? Am I going too fast? Nak perlu saya slow down ke?
Tak.
Okay. Good. Thank you for your response. Alright. 10.33 kita start. Stretching dulu. Okay, 10.33. Alright, let's go. Let's take a look at the next question. Okay, let's take a look at the next question. Okay. So, gambar ni menunjukkan reproduction by stem cutting because it tries to emulate vegetative reproduction atau asexual reproduction. So let's take a look here. Gambar GIF yang saya bagi dekat atas tu, okay, saya tunjukkan with rapid rooting powder tu is basically the hormone that we are talking about lah. Hormone ni sebenarnya commercially available. So macam bila ada pekebun atau petani yang berminat untuk buat vegetative reproduction atau asexual reproduction ni dengan cara stem cutting ni, dia boleh beli produk ni sebenarnya, hormon ni. Nama produk ni dipanggil ni adalah nama brand, Rapid Rooting Powder. Tapi dia basically ialah hormon yang kita nak bincang ni lah. So nama hormon dia, dia lukis gambar bendera pula. Nak berkarya pula kat sini ya. Macam-macam lah hampa ni. Okay. So tulis jawapan dulu sayangku. Tulis jawapan dulu. Satgi hampa berkarya. Okay. Satgi hampa berkarya. So number 1. What is the name of the hormone? We learn 2 types of hormones in our syllabus. So sama ada hormon A atau hormon G. What is the name? And then what is the function of that particular hormone? What does it do? What does it, whenever we talk about hormone, the presence of hormone is to stimulate atau to promote apa? Okay, kita selalu gunakan perkataan sama ada stimulate atau promote for the verb that we will be using. And then describe lagi apa lagi proses-proses yang berkaitan bila dah berlaku promote something tu menyebabkan apa kesan dia kepada roots. Roots jadi apa? Okay, promotes growth in plants too is a good, but is a good idea, but it is a general idea. Kena specify lagi. Promotes growth in what part of plant? Okay. Tak betulkan sikit, tambah sikit. Okay, cell elongation of plant cell. Specify lagi. Bukan plant cell. Sel mana? Sel mana? Kena specify lagi. What cell? Which type of cell? Okay, we have learned about types of cell in plant kan, dan sebenarnya hormon tu letaknya dekat bahagian bawah. So dia promote elongation of what type of cell? Okay, promote elongation of meristematic plant cell. Okay, good, good, good. Promote the growth root of plant cell. Good, good, good. Okay. Okay, so point number 1, sila tanda ya cikgu-cikgu. Point 1 is auxin hormone. Hampa sebut, hampa nyatakan nama hormon tu auxin. You mention that the name of hormone is auxin, then you got 1 mark. Okay. Point number 2 is promote growth of roots. Promote growth in roots boleh, promote root growth boleh, stimulate the growth of root boleh. Macam tu ya. Point number 2. Point number 3, bila kita habaq kata promote growth, mestilah kita boleh habaq proses, iaitu increase cell division atau increase mitosis in root cells. Okay, atau increase mitosis atau cell division in meristem. Meristem lagi tepat lah. Okay, number 3, point number 4, therefore more roots can be produced. Atau saya suka jawapan ni. Roots grow longer. Betul itu. Betul. Bagus ada Okay, roots grow longer. Betul. Saya setuju. Roots grow longer atau roots more roots produce, macam dalam gambar tu kan saya bagi more roots produce. Okay, and then kesan apabila lebih banyak root terhasil, apa kesan dia? Apa kerja yang root boleh buat dengan lebih baik? Iaitu more water can be absorbed atau more mineral can be absorbed. Kan? Okay. So, point 1, nama hormon, auxin. Point 2, kerja hormon tu promote formation atau promote the growth of roots. Point number 3, increase atau promote cell division atau mitosis. And then point 4, therefore more roots can be produced atau roots can grow longer. Bila roots dah grow longer atau roots ada lagi banyak, maka more water and minerals can be absorbed. Okay, any 3 out of 5 points that I've mentioned to you. Okay, kita ada Jerman, tim Jerman ada, tim Itali, tim Jerman dah hilang dah. Tinggal tim Itali saja. Masya Allah. Okay, tim nak lukis bendera Malaysia susah Ada banyak bintang, ada bintang yang banyak bucu. Alright, thank you so much for all of your responses. Okay, I can see that you have highlighted your answers. Alright. Okay, mari kita tengok soalan seterusnya. Ini soalan draw. Meh kita draw. Meh kita sama-sama draw. Okay. Kita sama-sama draw. Tadi nak draw kan? Kita bagi draw. Okay, how do you draw? How to draw? Okay. Ingat, sugar pula. Yelah, sugar. Deoxyribose sugar tu is a pentagon. Is a pentagon. So lukis pentagon. Lepas tu bucu kiri kanan pentagon tu, bucu kiri atau bucu kanan tak kisah. Bucu kiri kanan tu satu gabung dengan phosphate group. One binds with phosphate group. Another bucu binds with nitrogenous base. Okay, make sure you label because drawing is one mark, correct labeling is another mark. Tak ada label, macam ni tak ada label. Okay, gambar yang saya tengah pahami dalam Google Meet sekarang ni, gambar dia ni kena betulkan sikit. Make sure that the line connecting phosphate group yang bulat ni kena dengan bucu. Kena dengan bucu sugar, pentose sugar. Sama juga, nitrogenous base ni pastikan line dia kena dengan bucu. Okay, kena dengan bucu. Okay, macam apa? Macam Fatimah buat ni, okay, macam Fatimah buat ni, dari segi bucu betul dah. Tapi Fatimah kena bawa nitrogenous base tu pergi ke bawah, ke kanan. Sebab dia kena selari. Okay, okay. Untuk gambar gambar Ilhan ni, Ilhan kena bucu bawah Ilhan. Salah sambungan bucu tu. Okay, Fatimah tu bawa bucu tu, garis tu ke bawah sikit. Lepas tu, okay, kejap ya, mari kita tunjuk. Okay. Ini contoh jawapan daripada Amir. Okay, ini jawapan Amir. Jawapan Amir ni bagus. Jawapan Amir ni kedudukan phosphate group, the way you place the phosphate group is correct and the way you place the pentose sugar is also correct. So Fatimah cuba tengok cara Amir letak nitrogenous base tu. Dia kena selari. Kena ke bawahkan sikit nitrogenous base tu ya. Okay, good labeling. I can understand your labeling Amir, very good. Apa Fatimah punya labeling pun saya boleh nampak. Cuma kena betulkan ya nitrogenous base tu apa. Alright, itu adalah jawapan Amir tadi. Allahu Akbar. Macam jadi pameran gambar bendera pula ya. Okay. Okay, so Janisha Janisha your phosphate group tu kena betulkan. It should be left and right macam okay kejap ya. Ni ya, saya pamerkan kat sini. Ini contoh yang Mikael buat. Okey, nampak ya kedudukan tu, sambungan tu berlaku di kiri kanan bucu. Kiri kanan. Okey, bukan atas bawah. Janisha betulkan ya. Phosphate group, the position of your phosphate group tu kena betulkan sikit. Sikit je lagi. Okey, good. Bagus dia ambil tindakan terus. Alright. Okay, good labeling. I can understand your labeling. Make sure ada garis je lah Megat. Okay, kena ada garis ya. Tak apa, saya faham limitation untuk guna paddock ni. Especially untuk kawan-kawan kita yang baru first time guna kan. Sebab last time bila saya buat kelas dengan 5 Aurora, saya tak guna paddock ni rasanya lagi. Saya belum guna lagi. Okay, ini adalah jawapan Nur Izzati. Jawapan Nur Izzati pun okay, betul. Cuma saya nak minta Nur Izzati bawa turun sikit nitrogenous base tu. Bawa turun sikit macam Sabrina buat ni. Okay, macam Sabrina buat ni. Kedudukan nitrogenous base dia okay, good. Okay, kena ke bawahkan sikit ya Nur Izzati. Mari kita tengok siapa lagi. Alright, okay, hot ni. Okay, Muhammad Mikael, please take a look. Sekejap ya. Saya kena. Okay, please take a look at your answer. Cuba kita compare dengan yang kawan kita punya ni. Kamu punya kedudukan nitrogenous base tu tak berapa tepat. Muhammad Mikael tolong betulkan. Saya pamerkan dekat Google Meet supaya kamu boleh banding dengan kawan kamu punya dah. Okay, kedudukan garis sambungan dengan nitrogenous base tu kena betulkan. Alright. Okay, good. Okay, drawing settle. Okay, drawing settle. Okay, sekarang ni Mari kita tengok pula comparison. Okay, mari kita tengok pula comparison. Okay, I've given you the name of the nucleic acid. One is DNA, another one is RNA. Most of the differences are differences that can be observed there. For example, berapa benang atau how the number of strand between DNA and RNA. That is jangan tak tahu dulu sayangku. Cuba tengok diagram. Okay, take a look at DNA and RNA. The number of strand atau bilangan berbenang. Cuba tengok DNA ada berapa benang, RNA ada berapa benang. That is number one. Tak sebut double pun tak apa. Kamu sebut has two strands. RNA has one strand, for example. Okay, good. Bentuk dia satu helix, satu straight atau linear. Okay, dia punya dah ada dua difference dah. Tanya. You already have two differences. One is number of strand. Number two is the shape, whether it is helix or it is linear. Kan? So bila kita nak observe differences between structure, sebab dia bagi gambar. So we are soalan tu dia tak kata pun dia tak kata pun differences tu in terms of function ke in terms of structure. So kita bagilah differences in structure sebab kita nampak gambar dia. So gambar ada tunjuk ada dua benang. There are two strands on the DNA molecule. So we can say that DNA has double strand, mRNA has single strand atau two strand and single strand. Bagus. Okay. And then the second one is we can compare in terms of shape. We can see the shape. Where DNA is helix shape and mRNA is linear shape. Okay, sayangku. Ada seorang sayangku ini, dia cerita asing-asing. Mu jangan cerita asing-asing sayangku. Bila comparison, kena cerita berturut-turut. Siapa ni? Siapa ni? Saya. Ya, kejap. May saya highlight kejap ya. Saya nak bagi feedback. Okay. So, jawapan yang macam ni seperti yang saya pamer ni, ini bukan differences. Ini kamu hanya menyatakan sifat secara berasingan. So, there is no mark here if you write like this. Okay, cuba tengok dekat Google Meet. Okay, kena tulis, kamu nak cakap pasal strand. So, gabung. DNA ada dua strand, RNA ada satu strand. Satu statement yang menunjukkan dari ayat tu you are comparing. Macam tu. Dia tak boleh asing-asing ya. Bagus. Dia terus ambil tindakan. Habiskan. Yes. Tadi ada macam ada siapa-siapa nak tanya soalan ke? Boleh tanya? Do you have any questions? Okay. Difference seterusnya yang saya nyatakan lepas ni adalah differences yang kalau kamu memang hafal atau ingat fakta mengenai DNA atau RNA. The following differences are differences in terms of characteristics that is not observed here. So number one is DNA has deoxyribose sugar. mRNA has ribose sugar. Number one. Number two, DNA found in nucleus or chloroplast or mitochondria, whereas ribosome found in mRNA found in cytoplasm or ribosome. Number three, number four, the function of DNA is to store genetic information, whereas mRNA is involved in protein synthesis. And then difference number five is in terms of nitrogenous base. DNA has nitrogenous base thymine, whereas mRNA has nitrogenous base uracil. Okay. Alright. Saya copy and paste semua differences ni pada Google Chat dalam Meet ni. Alright. Tulis semua dalam Meet supaya kamu boleh rujuk kemudian. Alright. Let's go. It's already 10:50 anak-anak. Allah, 10:50 dah. Alright. So, in this question, I've given a diagram. Pada diagram tu, I've already show you the name of that particular protein, which is a tertiary protein. Pasal apa kita tahu tertiary protein? Because it is consists of beta pleated sheet and alpha helix. Alpha helix dengan beta pleated sheet adalah secondary structure for protein. So when there is a combination of secondary structure protein membentuk tertiary protein. Oh, boleh terus jawab? Tak apa, boleh terus jawab. So an example of alpha helix protein is hair keratin, whereas beta pleated sheet are used to form certain types of amino acids like valine and tyrosine. Tapi kita tak perlu hafal ya. Nak habaq mai saja. Okay, so tertiary protein is an example of enzyme. Tertiary protein, enzyme is a type of tertiary protein. So sebenarnya dia nak habaq mai kata, hampa boleh identify tak this particular tertiary protein is a type of enzyme, a type of enzyme. Okay, number one. So that should be your answer. Z is enzyme. Kalau hampa boleh habaq mai nama spesifik enzyme, that would be great. Okay, protein Z is enzyme. That is point one. Okay, in the picture, what I'm showing you in the slide is buah koko. Okay, buah koko ni kita actually ambil dia punya seeds, dia punya biji. Sebab tu kita panggil cocoa beans. So dia punya isi tu kita kena kita kena keluarkan isi yang warna putih tu. So isi yang warna putih tu sama macam isi buah, dia kaya dengan starch. So we want the beans, kita nak biji kat dalam dia. Orang Melayu panggil biji koko. Kita nak biji koko tu, kita tak nak dia punya isi. So we use enzyme to remove dia punya the fleshy part of this cocoa. We want to remove the starch surface of this cocoa on the cocoa beans. And then barulah lepas tu kita keringkan. Lepas tu dapatlah cocoa beans yang macam dalam gambar. Okay. So kalau kita punya okay, kita tahu tertiary protein tu is enzyme, number one. Number two, kita tahu kita nak break down starch atau remove starch. So there are 2 points already there. P is enzyme atau P is amylase. Kenapa amylase? Sebab we want to remove starch. Kita kena ingat kalau dia punya substrate is starch, enzyme dia starches pula, amylase. Okay, kalau tulis amylase boleh, tulis enzyme pun boleh. So P is enzyme, the protein is enzyme or amylase. Point number 2, used to digest starch atau remove starch. Okay, and then bila dah digest starch tu converted to become maltose atau disaccharides. Okay, any 2 out of 3 points. Okay, so enzyme remove the starch. Starch ya? Stretch pula. Starch. S-T-A-R-C-H. Oh, atas tu tersasul tarik kot, tak apa. Okay, and then break down starch to become maltose or disaccharide. Very good. Alright. Okay. Okay, let's take a look at the next question. Next question is the application level. Kenapa application level? Sebab it discusses about factors influencing enzyme activity. So saya bagi 2 extra stimulus atau 2 extra hints kat situ. Number 1, saya bagi gambar diagram, gambar graph the effect of temperature towards increasing enzyme activity. The question said the employee set the temperature to 56 degrees Celsius. Look at the graph. Is the 56 degrees Celsius the optimum temperature for the enzyme activity? What happens when the temperature is not the optimum temperature? So gambar kedua kat bawah tu, I'm showing you the effect of setting the temperature at 56 degrees Celsius towards protein Z. Okay, the term should be there, and then the effect on enzyme reaction kena specify dengan lebih detail lagi. Effect on enzyme reaction kena specify dengan lebih detail lagi ya. Okay, I'm showing you the responses of your friends. Okay, good. Okay, good. Okay, so point number 1, saya tak kisah kalau kamu punya tertib tu tak sama dengan saya, as long as the logical sequence is there. Okay, nombor 1 untuk saya, approach saya ialah saya habaq mai dulu 56 degree Celsius tu optimum temperature ke tak? Kalau bukan optimum temperature, lebih ke kurang daripada optimum temperature? My first point would be I will compare the temperature from the question. I will compare it to the optimum temperature of enzyme. Is it more or less than optimum temperature? That is my point number 1. So point number 1, I would say the temperature set by the factory worker atau the temperature set by the employee is more than optimum temperature. Okay, and point number 2, I would mention, saya akan habaq mai kesan temperature yang tinggi tu terhadap protein. Saya akan habaq menggunakan istilah dia iaitu denaturation atau denatured tu. So the point number 2, I would say because of therefore the enzyme will become denatured. Okay, therefore enzyme will become denatured. Kamu tulis enzyme tu jadi denatured, dah dapat markah. Atau enzyme change shape. Saya boleh terima juga. Okay, when the enzyme is denatured, dah change shape, it will not be able to bind with starch. Atau it will not be able to bind with substrate. That is point number 3. Point number 4, ada tak nak habaq pasal active site? Boleh. You can say active site changes. Bila kamu tak sebut denatured tapi kamu sebut active site dia berubah. Boleh ya. Okay. And then number 5, kena simpulkan dengan kesan terhadap factory tersebut. Make chocolate manufacturing become slower atau chocolate manufacturing become incomplete. Okay. Atau rate of enzyme reaction will become lower. Okay, saya terima. Rate of reaction decreases pun saya terima. Okay, any 3 out of 5 points that I've mentioned. Alright. Okay, so this is a type of KBAT question. Walaupun dia bagi soalan tu dalam konteks aplikasi dalam kilang, tapi sebenarnya benda yang kita guna, maklumat yang kita guna, konsep yang kita guna untuk explain adalah konsep yang kita belajar. Tengok, saya bagi gambar graf ni memang kita ada belajar. And then denaturation ni pun is a process that we already covered. Alright? So hopefully don't be afraid with soalan, this kind of questions. As long as you can digest the information, then it's okay. Alright, kita bertenang sikit pada slide yang seterusnya sebab saya bagi soalan multiple choice question. Multiple choice question. So the function of R tu saya dah label kat situ ialah I think it's the afferent neuron. Okay, yes. R2 afferent neuron. So which one is the function of afferent neuron? Okay. R2 pada soalan, kalau kamu rujuk balik pada kamu punya hard copy, sebab saya dah cover pula dekat situ. R2 ialah afferent neuron. Saya dah cover kat situ Sorry. If you are confused, sorry. Okay, let's take a look at your responses. It is the function. Okay, okay, sebab ini adalah soalan state the function Okay, so the diagram shows R is afferent neuron. So the function is A. Pilihan jawapan A, okay? Pilihan jawapan yang pertama. Okay, saya move on sebab ini very simple saja. If you can state atau menyatakan fungsi ni, you got it already. Okay, let's take a look at the next slide. Okay, pada Google Meet, sekejap saya tak pasti GIF ni jalan dekat paddock tu. Oh, dekat paddock dia tak jalan. Dekat Google Meet, saya lampirkan dekat situ a GIF image showing from the receptor. Okay, dalam mata tu ada receptor. Nampak bila yang masuk tu ialah gelombang biru, reseptor biru yang akan activated, gelombang merah, reseptor merah akan activated, and then garis if you want to answer, you can directly answer sambil-sambil saya cerita tu boleh terus answer, no problem. So the pathway from the eyes to the brain is the afferent neuron. Okay, the pathway from the between the eyes to the brain is the afferent neuron. So saya tunjuk GIF kat situ, saya nak tunjuk how these steps occur lah. Okay, so macam biasa, bila kita cerita reaction macam ni, kita cerita peranan reseptor dulu. Kita cerita peranan reseptor dulu. Okay. Number one, okay. Reseptor buat apa? Number two, number one, reseptor menghasilkan apa? Number two, hasil daripada reseptor tu, apa yang afferent neuron buat? Point number three, what happens to the information sent by the afferent neuron? Itu final dia, final destination dia. And then, what happens in the brain? What does the brain do? Okay, what does the brain do with the information from the sensory? Wah, ada orang dah macam dah tak jawab dah banyak nak type Allahu Akbar. Ramai ni Fana pun dah tak jawab dah. Tak apa, I understand. Cepat sangat sorry Janisha, am I going too fast? Tak apa, it's okay. Okay, you can check out the video once again if you want to take a look at your friend's answer. Mungkin lepas ni boleh rujuk video sambil rujuk notes, paddock notes ni Okay, number one, point one, receptor in the eye detect the stimulus. Okay, atau receptor in the eye will generate nerve impulse. Okay, receptor in the eyes. Saya kurang setuju untuk statement the sensory organ detect. Sensory organ tu terlalu besar. Kena sebut receptor. Kena sebut receptor. Kita mencari istilah receptor tu. Okay. Receptor in the eyes detect the stimulus and produce atau generate nerve impulse. That is point number one. Point number two, nerve impulse transmitted along afferent neuron. Atau afferent neuron will transport nerve impulse to the interneuron. Okay. Nerve impulse transmit impulse along afferent neuron, atau afferent neuron transport impulse to interneuron. Okay. And then the next point is impulse transmitted to the brain atau transmitted to the interneuron. Final point, brain will integrate atau interpret the information received. Integrate atau interpret the information received. Okay, so ada kawan kita dia statement yang sebut the eyes detect. Tukar kepada receptor in the eyes detect. Tambah instead of the eyes detect, jawapan kita kena lebih spesifik, receptor in the eyes detect. Good. The stimulus will be detected by photoreceptor. Good. It is a good introduction. It's a good first point. Good. Okay. Alright. I give you one more minute. 11:07. Kita pergi ke next question. Okay. Rasanya kita sampai soalan 4 je ni. Okay. Okay, 11:07. Sorry, I have to move towards the next slide. Okay. So, untuk soalan ini, describe what will happen to the action. Semuanya ini adalah contoh jawapan soalan in negative responses. Kita hafal fungsi setiap satu structure dalam ni. We know the function of the components in the diagram, but we change it to a negative form. No impulse will send to the okay, yang ni tak apa saya bagi, tak apa. No impulse sent to the effector, number 1, atau no impulse sent to bicep, point number 1. Point number 2, therefore, bicep does not contract atau muscle does not contract. Point number 3, no response atau movement of the arm. Semua no. No impulse sent to bicep atau no impulse sent to effector atau no impulse sent to the muscle. Point number 1. Point number 2, effector atau muscle does not contract. Point number 2. Point number 3, therefore there is no response. There will be no response atau movement of the arm. No impulse, no contract, no response. Okay. No impulse, no contract, no response. Alright. Okay. Alright, it's okay. I truly understand the limitations of this particular kenapa sedih Amir? Kenapa nganga mulut Amir? Are you okay?
Sedih.
Oh, okay. Aduh, sabar je lah. Alright. Okay, I will do my best to the best of my ability. Saya akan cuba screenshot all sample answers that your friends have given. Nanti sample answers ni saya combine, saya hantar dalam bentuk PDF ya. Because I cannot share with you the copy of mark scheme sebab dia under copyright laws. So kalau mark scheme tu diubah suai dalam bentuk jawapan-jawapan pelajar macam ni, I can share this one. Okay, I can share this one. I cannot share the actual mark scheme. Dia ada copyright laws. SBP ni dia ada detail sikit pasal benda ni. Okay, I think this is the last one. Okay, this is the last question for question 4. Okay, saya ada bagi clue dekat atas tu, apakah differences yang kita boleh cerita. 4.1 is a type of voluntary action because we have to think and plan before we do that. We do that particular action. But 4.2 shows involuntary action which we does not we are not consciously controlling this particular actions. Okay. That should be difference number one. The type voluntary versus involuntary action. 4.1 voluntary, 4.2 involuntary. Okay. Kemudian siapa yang control? Kalau voluntary action, bahagian mana yang control? Voluntary action is controlled by cerebrum. Involuntary action controlled by medulla oblongata. Okay, saya setuju dengan jawapan kawan kamu ni. 4.2 reaction dia lambat, the reaction is slow. 4.1 the reaction is fast. Good. Bagus. Mari kita tengok idea lain. Kita dah ada 3 difference ya. Kecepatan, jenis dan controlled by who. Okay. Okay, and then types of effector. Effector yang terlibat pun boleh. 4.1 effector dia ialah skeletal muscle. 4.2 effector dia ialah smooth muscle. Okay. Okay. You already got 3 differences. Good. Teruskan. Okay, and then okay lah, saya rasa cukup kot. Okay, sorry anak-anak, saya terpaksa berhenti pada soalan 4. Saya terpaksa berhenti pada soalan 4 sebab 11:15 is the end of our class. Directly after this, I have a class with Form 4 students. Okay, but I think your answers, your differences are good. Okay, so nanti I think we have about a few days before paper bio that we can continue this discussion. Okay, so last kali saya tahu I ask you a lot, but this is important to me. Class atau lesson reflection. What did you like about today's lesson? What didn't you like? What was easy? What was hard? For what was hard, if you have any reaction, concept that needs re-explaining, please put it in what was hard. Okay. Kalau macam saya ada laju ke in terms of the way I deliver the content, ada masalah, put in under what didn't you like. Okay. Question very hard. Memang question very. Tapi sebenarnya panel yang buat soalan SBP ni I know them personally and most of us are trained by lembaga peperiksaan. So soalan dia agak standard sebenarnya. Okay. Oh, suka drawing pantai. Okay. Okay, suka time too short. Okay lah, tak apa. Next. Saya memang ideally, saya prefer kelas yang sejam setengah kan, tapi nak buat macam mana kan. Tak apa. It's okay. We work within our confine kan. The answers are too detailed. Okay. The answers are too detailed tu tak boleh buat apalah. That is the nature of bio question. We have to work on that lah. Okay, if you think the answers are too detailed, okay, we have to work on that. Make it, I will help you to understand why we have detailed answer. Yelah, nak jadi scientist kan. Okay. Coordination is pretty cool. Anything related. You know what? Your feedback about what was hard, anything related to plants kan? Many students before this also have the same response or the same feedback. So if you take a look at the current syllabus, bio syllabus, all Form 4 chapters are regarding humans and animal. All Form 5 chapters are regarding plants. So memang tak boleh lari lah. Masa form 5 belajar plants saja untuk KSSM ni. Okay. Anaphase-nya importance. Anaphase punya importance ya. Okay. Tapi kita sekarang kan kita jawab soalan structured question saja. Kita buat structured question saja. Nanti tanya 5 solaris hari tu kita buat soalan essay lagi panjang kan. Kita buat soalan 8 markah, 10 markah. Pakai pedek je. Tapi sebab mereka dah lama guna pedek, so mereka dah expert guna pedek. Okay, so thank you so much for all of your responses. Saya dapat memahami kualiti pengajaran saya dengan baik, dapat menilai dengan baik. Okey, sekejap ya. Saya minta maaf sekali lagi kerana saya tidak buka kamera sebab saya sebenarnya sakit mata dan although it is healing, my eyes are still hideous, so I don't want to show my eyes to you. Sekejap ya, saya nak ambil gambar untuk attendance saja sekejap ya. Saya terlupa nak ambil attendance awal-awal tadi, so I have to take a picture. Okay. Okay ya, kejap ya. I take picture first and then we can go lah. Okay. So, sekejap ya. Saya take picture. Okay. And then, okay, that is all for today. Thank you so much for coming. May Allah bless all of our efforts on this blessed day. Thank you.
Terima
Thank you,
kasih
teacher.
cikgu.
Sama-sama.
Thank you, teacher.
Thank you, cikgu.
Thank you.
Assalamualaikum.
Thank you, teacher.
Thank you, teacher.
Thank you, cikgu.
Thank you, madam.
Yeah, you're welcome. Alamak.
Thank you.
Thank you, madam.
You're welcome. Okay, nanti saya bagi recordingnya. No worries. Kejap. Kena habak my dekat form 4 ni saya masuk lambat.
Thank you, cikgu.
Sama-sama. Serita, Aisyah Dahia dah habis dah?